hopefully, some of you may have noticed the tall, good-looking younger guy who was standing up here before. He's your guest lecturer for Friday. He's sitting over there in the corner, trying to be as unobtrusive as possible. Their lectures, hopefully you noticed the first lecture was recorded, was online. Unfortunately, the one for Friday won't be. But yes, there will still be questions on the exam. Yes, there will be clicker questions. No, they won't count. And I've had a couple of people ask me, I haven't posted on D2L, but I'll do that right after class, how to register your clickers. You can either use your username or your student ID. For those of you who've done student ID, that's fine. You could also just use your Odin login username, because that's how it's actually going to be connected through D2L eventually. So speaking of clicker questions, here's our first one. From a molecular point of view, there's the most diversity in A, bacteria, B, animals, C, plants, D, fungi, E, humans. And we'll actually start the clickers here eventually. Once I get the, again, technology always takes about a week for us to get running on these things. So come on, go. Start. Start. Is it actually going to start? No, it's still not going to start. Come on. A through E. See, I promised the technology would work, and now it isn't. Very strange. My probably on the wrong system here. Well, so clickers are not working. So we can just do a vote of hands. It doesn't count anyway, right? So how many think bacteria? How many think anything else? Good. This one's actually a little bit more challenging. Which of the following is the best description of genes that are in a gene family? They are homologs, they are paralogs, they are orthologs, they are paradoxes. They have identical sequences. I'll let you think about it for a second or two while I try and get this actually going. Yay, it's actually now working. Go ahead and vote with your clickers. Now that you put them all away. I'll give you a minute. Okay, I'm sure there are more than 70 people in this class. I haven't counted you, but I'm pretty sure there are. So, what do you think? Oops. Pull this over here. No one thinks they're paradoxes. Come up with some other answer for these things. Certainly don't have identical sequences, and I'm actually really glad that people didn't put that down. We've got then homologs, paralogs, and orthologs. Homologs include both paralogs and orthologs. And so actually that's a more general term, and that just says they have a what? They've come from the same source. They have the similar history, which may or may not be detectable by sequence similarity, which we'll take a look at as we move along through the rest of today. So, yes, they are homologs. They could be paralogs, they could be orthologs, but they're definitely going to be homologs of each other. Okay, so quickly in review, last time we just talked about the central dogma. i.e. DNA goes to RNA, goes to protein. And again, that's basically what molecular biology is all about. That's what we're going to be talking about for the next 9.5 weeks in terms of how that process takes place. We'll look a little bit at genomes. This, again, could be a whole course in its own right. But important as far as this course is concerned is that you've got lots of different sizes of genomes. There's protein coding sequences, actually relatively small numbers of protein coding sequences in our genomes. In bacteria, there are much more protein coding sequences that are present in the genomes. And it's a lot of the regulation of what parts of those genomes get turned on or turned off, which is really important for what makes us us and what makes bacteria bacteria. But also, together with that idea of genomes, there's a lot of things that are really similar across all cellular life as we know it. And so it's that commonality which we're also going to be talking about quite a bit. Again, we mentioned gene families. Gene families are now those sequences which are going to encode something which has, again, developed from a common ancestor. It could either be ortholog or paralog, i.e. it could be something which is being duplicated in one organism and then diversifying in that organism, or be paralog, so it's going to be diverging in different organisms. But still, each of those eventual genes is similar to each other because it has a common ancestor. And so those are how you get gene families. And if you look at genomes, going back to genomes, that's a real big part of our genome, and even a lot of the smaller, simpler genomes. A lot of these are present as gene families. Then we talked about model organisms. Sort of briefly at the end, the idea of model organisms is, again, talking about this commonality, thinking about things that are the same or at least very similar between all different kinds of life. We can't study all different kinds of life, so we pick and choose the ones that are best suited to the studies that we want to work on. So if we're interested in bacteria, most of the time we work with E. coli. It's not true of all bacteria, is that first question. So way more molecular diversity in bacteria than there are in all animals, or for that matter, animals, plants, and fungi. But at least anything that's similar in bacteria to, as I believe it was François Jacob said, if it's true for an elephant, it's true for E. coli. Or if it's true for E. coli, it's true for the elephant. And so those are the kinds of real fundamental aspects that we want to talk about. The other aspect about model organisms that's really important is they're usually genetic tools for these organisms. So there's the geneticists I mentioned right at the end. What they'd like to do, like to break things and then try and figure out what they broke. And so that's really a critical way of understanding how things work. Today, we'll cover a year of chemistry and a year of biochemistry in one lecture, maybe. But these are the important things that we need to know as far as this course is concerned from a chemistry point of view. We'll talk a little bit about atoms, mostly about weak interactions, because it's the weak interactions that are most important for molecular biology. And it turns out, not surprisingly, these weak interactions, you need a whole bunch of them. And that gives you the flexibility that you need in biology, but also the reversibility. Everything we know about biology has to do with water. That's partly why NASA is looking for water on other planets, because water is absolutely critical to all of the biological chemistry that we know about. If it wasn't for water, we wouldn't be here. And this planet probably wouldn't have life, etc. One of the reasons that I'll talk about is in terms of hydrophobic interactions, but that's by no means the only kind of important aspect of water that's here. Then we'll talk a little bit about some strong interactions. These are basically covalent bonds and how you make covalent bonds. That process really has to do with activation energies. So we'll talk a little bit about thermodynamics. Again, very small amount about thermodynamics here. Hopefully, this is all review. Then the high energy bonds. These are the even higher energy covalent bonds. And then coupling and precursor activation. And then we'll very briefly cover protein structure. What atoms do we need to care about? Fortunately, most of the periodic table down here in gray at the bottom, you don't need to worry about. It's really about these pink atoms here, hydrogen, carbon, nitrogen, oxygen. 90+% of all biology and is involved with these particular atoms. You've got a couple of ions, particularly chloride, sodium, magnesium, potassium, and calcium. And we'll talk about phosphorus towards the end here. Phosphorus is really nice because it has this great charge, at least in aqueous environment. It's a phosphate that allows you to change things, particularly in terms of structure, really rapidly. And sulfur, which also provides nice extra bonding kinds of activity. So speaking of bonds, in the textbook they mention two kinds of bonds. I don't like this ionic bond here at all. I like to think of this much more as an ionic interaction. But certainly covalent bonds, all the covalent bonds are sharing of electrons between two different atoms. And we're not going to get into the quantum chemistry of this, but basically, again, it's these electrons which are being shared between two nuclei of the different atoms, and this gives you a very strong interaction. These things are going to stay together. The atoms are going to stay together really quite tightly relative to each other. You can also have ionic, again, as I say, interactions, and that's literally the transfer of an electron from one atom to another atom, and that process is, of course, going to give you differential charges. And so now it's going to be an electrostatic interaction between these two atoms as opposed to a sharing of electrons. What are the energies of these things? Previous version of the textbook, interestingly, had these in kcals per mole. Now we're in kilojoules. There's a pretty, I think it's what, 4.8, something like that, conversion factor. So if you like kcals, it doesn't really matter. Kilojoules is SI, so we're going to use kilojoules. Covalent bonds, again, these are these shared electrons, about 350 kilojoules per mole. No, I don't expect you to remember these exact numbers. What I do expect you to remember, however, is that this number, 350, is a lot more than average thermal energy. And the average thermal energy is just what stuff is bouncing around. When I say average thermal energy, it's at 37 degrees Celsius. Our lab works at 80 degrees Celsius, most of our organisms, so very different kinds of thermal energies there, but still much less than what you have in terms of covalent bonds. On the other hand, those weaker bonds that I mentioned before, the ones that are really important for molecular biology, are much weaker and on the order of the average thermal energy. So basically, one of these bonds or one of these interactions is not going to be strong enough to hold two molecules or atoms or whatever it is together. You have to have lots of them. And lots of them, from a biological point of view, is really nice because it allows you specificity. You have to have many of these kinds of interactions. And it also allows reversibility. It allows you to change that. And was it Buddha who said there's nothing constant but change? Also extremely true in biology as well. So we'll go through and talk about these individual bonds again, or interactions. Again, critical here, you've got to have a whole bunch of them to have them actually work, and they come and go. Briefly want to talk about polarity versus polar. Again, we've talked about the molecular biology polarity. You've got a front end and a back end of something. In terms of chemical polarity, we're talking about having slight positive and slight negative charges. Probably, again, the reason we have life on Earth is because of the chemical polarity of water. It's not the same at both ends, unlike your oxygen molecule. It's got a slight negative charge here and slight positive charges down here. Now, of course, what that means is that water is really good at interacting with itself, but also really good at interacting with lots of other atoms and molecules. Classic example here are these so-called hydrogen bonds, and this is, all it really is now, instead of sharing that electron, which you had in terms of a covalent bond, now you're basically sort of sharing a hydrogen atom between two different other atoms. This could either be two oxygen atoms, oxygen-nitrogen, nitrogen-oxygen, nitrogen-nitrogen. And all that this means is that these things are associated with each other because of this slight separation of charge. So now we're going to have a negative oxygen here, your positive hydrogen. Those are going to be aligned with each other and then have an attractive force between them. And so that's your hydrogen bond between them. And again, these are relatively weak interactions. One of them is going to be nowhere near enough. You need to have lots of them or combinations with other interactions in order to get a stable interaction between the two. Another one of these weak interactions are the so-called van der Waals interactions. This has much more to do with the electronic state of atoms. I'm not going to get into why, at least not in terms of this course is concerned. But basically, it's that you get two atoms at just the right distance away from each other. So just like the three bears, not too big, not too small, just right. And that just right distance away from each other, which is shown right here, is going to then give you attractive interactions between those atoms. Very weak attractive interactions between those atoms. And as soon as you move away from this very short distance here, you get big changes. And so, for instance, here, if you start to get closer than those van der Waals attractive interactions here, you notice this becomes repulsive extremely quickly. So again, it's really that sweet spot. You've got to be in just the right place to have these van der Waals interactions be, attractive to each other, and they drop really quickly. So as soon as you get further away, that attraction is really very, very low. Again, these are very small in terms of their actual amounts. Hydrophobic interactions, lots of people talk about hydrophobic bonds, etc. I just like to think of hydrophobic interactions as really great hydrogen bonding that forms between water molecules. And so basically, all that hydrophobic interactions are giving you more hydrogen bonds that form between water molecules. So if you just look at the example here on the right-hand side, up at the top here, you've got hydrogen bonds forming between the water molecules. They're these little yellow bars here. Two molecules here that are not interacting with those water molecules. If those two now interact with each other, you have more hydrogen bonds that are forming between the water on the outside. So people also think about this as cages. I think in your textbook it also has a picture of a cage of these water molecules around these hydrophobic interactions. But basically, again, it's really the flip side of hydrogen bonding between water, which is basically pushing together all those things that are not being bound to by the water. So it's also known as the polar interactions. So as an overview, that's really what molecular biology interactions are about. Lots of weak interactions, here just shown as positive and minuses. Here we've got hydrogen bonds, we've got ionic interactions, van der Waals interactions as well. And it's all about how two molecules fit together, and fit together, particularly if you're talking about van der Waals interactions, in a very close and precise way. There's a animation about this that's connected to your textbook. I'm not quite sure how to get to it from the textbook. I think it's a website. But talks a little bit about these kinds of interactions and the bonds that go between them. I'm not that wild about it, but sometimes it's useful to look at that way. So now I want to digress a little bit, and again, get back to hopefully general chemistry for you. Talk a little bit about acids and bases because they're really important in terms of some of the amino acids we're going to be talking about, particularly amino acid side chains for protein structures. all that we need to know about acids and bases is basically they're giving up hydrogen atoms or picking up hydrogen atoms together with electrons. So if you look at something like acetic acid that we have up here at the top, COOH, together with water, because we're all in aqueous environments, all biology is aqueous environments, this molecule has a tendency to transfer this hydrogen to water, generating these hydronium ions. So it gives you a stable negative charge on this particular molecule. And that's that stable negative charge at a particular concentration of hydronium ions. And so depending on how much of this free water, i.e. not ionized water, is around, you will lose or gain this particular charge that you have on your other molecule. Most cellular systems are pretty close to pH 7, so almost equal amounts of these uncharged and ionized amounts of water which is there. And this is another way of looking at that. Thinking about water, it can either be hydronium ions or hydroxyl ions. So now let's switch gears a little bit and talk about what we're made of, what all cellular life is made of, and basically it's this set of 5 things over here, although ATP should really be sort of small molecules, electron carriers, etc. We talked about nucleic acids and proteins before. We'll talk much more about proteins later on today. But the idea really is here that there are macromolecules, again, that we talked about, the nucleic acids, the proteins, most of the polysaccharides and the lipids, and they all come from these smaller building blocks that then get assembled into macromolecules. And hopefully this should be pretty obvious. This is chemically hard to do, and so we'll talk about how that happens on a chemical basis the next 10-15 minutes or so. So a reminder, we are mostly water. People argue about whether it's 70% or 80%, etc. So vast majority of what's in any cell, any cellular life that we know of, is water. Of the 30%, 20-30%, which are actually not water, the most of that is protein. We've got quite a lot of RNA and DNA, less DNA than you have of RNA, and then some of these other things as well. So we're really going to concentrate on protein and then the polynucleotides, RNA and DNA here. But these are macromolecules. How do you make macromolecules? Well, not surprisingly, you use water, because that's what most of us are, and that's what's really critical for all of these chemical reactions. So basically, with maybe a couple of exceptions, but the vast majority of the chemistry we're going to talk about in terms of molecular biology has to do with either making water or using water. And not surprisingly, we're mostly water, so making water is hard, and using water is easy. Also, we can talk about these as condensation reactions or hydrolysis reactions. So adding water is hydrolysis, taking away water is condensation. Almost all polymerizations here, going from monomers to multimers. This particular case is DNA, but it's true for RNA, it's true for proteins, it's true for polysaccharides, it's true for most lipids as well. You have monomers becoming polymers through these condensation reactions. So loss of water to do that. Well, okay, that's great, but as I mentioned before, this is hard to do on a chemical process. And the reason for that is that almost all of these reactions, and when I say most biological reactions, that's mostly going to be condensation reactions, they are energetically really unfavorable. And so they're not going to happen. Well, we exist. We're here, right? We're having this class. So they must have happened. And so how do you get that to happen? And that's because these reactions are all linked to each other to give you a pathway. And so this is a classic sort of chemical progression diagram here. Here's our energy. We want to go from A to B, but to get from A to B, we've also got to go from B to C. To go from C to D is unfavorable, so we've got to go to E as well. A to E is favorable, whereas A to B and C or D are not. And so you can think of these as, for instance, a polymerization reaction, A to B, polymerization reaction, C to D, but somehow you've got to have these B to C reactions and the D to E reactions to make this actually take place. So one way that this happens, where do you get that energy from? Well, most of the energy comes from the sun. We just take advantage of that by eating things that have had solar energy, that they've been converted into something that we can use. Take these, now they become a useful form of energy, i.e. something that we can do to go from B to C, for instance, to drive A to B. And that pathway will take us from these monomers down here at the bottom to the polymers, which are what we need. Now, why are these kinds of interactions, or I should say reactions, favorable versus unfavorable? This is one of my favorite figures ever in a textbook, maybe because I'm a dad and I have 2 kids, but I think this is really sort of indicative of the second law of thermodynamics. If you leave something around and don't input any energy in it, it starts to look like this over here. Can you guys see that? But yes, this is what I have to deal with on a more or less daily basis. The only way to get from here back over to here is inputting some kind of energy. And I'll leave it to you to decide what kind of energy that is. I'm sure we all have different ways of inputting that kind of energy. But it's exactly true for this kind of case. Entropy is going to increase. There's going to be more disorder unless you put in some kind of extra energy, some kind of extra source of energy. And so the way that happens in terms of biological reactions is you have coupling. You have one reaction which is energetically favorable, which can then drive a reaction which is energetically unfavorable. So allowance, for instance. So that allowance helps you drive this otherwise unfavorable reaction. Input of energy, okay, allowance might not be the best way. You can think of some kinetic energy that you'd have to input too, but we won't go there. Not in my family anyway. But that's the whole idea, is that we have a energetically unfavorable reaction. This is the one that we want. Also known as a positive change in the free energy. So talk about delta G's. That positive delta G, that's not going to happen spontaneously. You have to have a spontaneous reaction, one which is going to generate energy, and it has to generate more energy than this reaction, which is having a different sign, is going in a different direction. So again, as most of you hopefully know, it's all about the ATP or the NADH, etc., and changing those kinds of amounts of energy. So polymerization, again, this is something, it's all about water, it's all about condensation reactions. Look at polysaccharides up here. Loss of water, condensation. Protein polymerization, loss of water. Nucleic acid polymerization, loss of water. So how do you get this energetically unfavorable reaction to take place? We have to couple it to some other reaction which is energetically favorable. And that brings us to our so-called high-energy bonds. And these are those that when you break them, and almost always that breakage of the bond is going to be what? It's biology. What's in hydrolysis? Exactly. It's going to be hydrolysis. So hydrolysis and condensation are almost always linked to each other in terms of this. So the hydrolysis of these high-energy bonds can then allow condensation to take place to give you these macromolecules. I have down here also practically irreversible. Well, this is a reminder to actually kind of go back and think about that image that we had of the room being cleaned up and the room not being cleaned up, having to do with entropy. If you've got two molecules attached to each other, that's pretty low entropy, right? If they're separated from each other, that's much higher entropy. So putting things together takes a lot of energy, but you've got to make sure that that process, again, it's going to be entropically really hard putting these things together, doesn't reverse itself. And so there are a number of different ways, and we'll talk about them particularly in terms of nucleic acid polymerization, to make sure that those reactions don't spontaneously fall back apart, because hydrolysis is way more energetically favorable than condensation taking place. Backing off a little bit, how do we get this energy? Again, I mentioned before, it's almost all about the sun. There are, again, all those in microbiology say, Dr. Stedman, you're so totally wrong. There's chemolithoautotrophs. You can't talk about this. Yes, there's other ways as well. But as far as we're concerned, most of this energy comes from sunlight, and it's all about photosynthesis. So it's taking water, generating oxygen, taking CO2, and generating organic molecules. Then we, being most living organisms, use this. We take the sugars and other organic molecules, convert them to CO2, and take oxygen and convert it to water. It's nice because this gives us a nice cycle, and basically, that's the chemistry of life, is fixation of carbon, usage of carbon, etc. It's also important to think about the concept of oxidation and reduction. I always had trouble remembering this, and then I went and sat in on Dr. Wamser's organic chemistry lecture, and he told me about LEO, and it's like, wow, that makes this so much easier. So what's LEO stand for? Loss of electron is oxidation. So that's all you really need to remember about oxidation and reduction. So we can look at that here, just looking at this covalent bond formation. Now you have this atom, which is losing an electron to this atom. This electron is more associated with this atom than this one, and so now this atom is oxidized. That process, losing electron, what kind of a charge does an electron have? Negative, so we end up here with a partially positively charged. So anything which is oxidized here is going to give you that partial positive charge. You can also look at oxidation and reduction on an organic chemistry scale. Very often, these electrons are also associated with hydrogen. So here, methane up at the very top. Here, the carbon, and we're just looking at the carbon here, is maximally reduced. It's got the most electrons. And as you move through to carbon dioxide, those electrons are getting further and further away from that carbon atom. So now that carbon atom has become mostly oxidized down at the bottom here. And well, more important actually for microbiology purposes and for cell biology, but it's this oxidation versus reduction and the transfer of electrons, which also can lead to formation of all of the nice, useful, high-energy bonds that we're mostly going to talk about for the rest of this course. But in terms of thinking about physiology, cell biology, etc., it's this reduction and oxidation which are really critical for that purpose. Now I need to touch on chemical equilibrium. Again, hopefully this is all review for everybody. If you have two sets of molecules here, could be atoms, etc., if they're in equilibrium, that means that we've got certain amount in one state and a certain amount in another state. There are rates between the two, how they convert between each other. So here we have a forward rate and a reverse rate, and then an equilibrium constant, which is this definition is true for all reactions, all interactions of different molecules, which is just the ratio of the forward reaction to the reverse reaction. So the rates are, not surprisingly, and they have a nice discussion of this in the textbook, proportional to the concentration of whatever things you have. And so you can also rewrite this equilibrium constant as the ratio of the product of your products versus the product of your reactants. So here, two reactants, reactant 1 and reactant 2, concentrations thereof. They're going to be your denominator, your numerator. It's going to be the product here on the other side. In this case, it's giving you 2. We could also just have 1, which is here. Why do we care about equilibrium constants? Well, at least as far as molecular biology is concerned, the main reason we care about equilibrium constants is not so much in chemical reactions. It's much more in terms of bonding and binding interactions of things that happen with each other. And so this is a way of looking at those kinds of reactions. All it is binding of one molecule to another molecule. Usually this is going to be a protein in terms of molecular biology, binding to something else, often a small molecule, sometimes another protein, very often a nucleic acid. And that will be called the ligand. Here it's ligand A. We've got multiple different ligands in this particular image. So if we think about the equilibrium constant, it's about the concentration of ligand times the concentration of product divided by the complex when the concentration of the complex when these two are actually together. So that's this reaction. Ligand plus protein to complex. You can also look at this in the same kind of way as rate constants. It's the rate constant of things coming apart from each other versus going back together. And so these are the two ways of expressing your K, equilibrium constant. When people talk about ligand protein or ligand-ligand interactions, they talk about a dissociation constant. So that's Kd. There are a couple of ways of looking at this. I like to look at it in terms of thinking about these two rates. So if the dissociation rate is a lot faster than the association rate, you're not going to have very tight binding because these things are going to be falling apart all the time, right? Because that's much faster. However, if the association rate is much higher than the dissociation rate, then these things are going to be together much more often. And so just by looking at the numbers down here, a millimolar association constant is going to be very weak interactions. A nanomolar dissociation constants can be very strong interactions. And if you think about these kinds of molecular biology interactions that are going on inside the cell, they could be enzymatic reactions, which we'll talk about in just a second. They could just be DNA-protein interactions. If you want those to happen, you want them to happen and be stable, you're going to want to have very low dissociation constants. You can also look at this in terms of back calculating. The KD is going to be the concentration of your free ligand when half of the protein is bound, because that will be, LP will be half of what L is. Yeah?
This slide is a little bit difficult for me to understand as far as the image. So
yeah.
Is that an example of like a ribosome or something building a... Okay, so what is L and P in that image? Like, how can I associate?
Okay, so yeah, so I'm sorry about the image is one that I expect from the previous textbook. So the question here is, what are we looking at? Yeah. And as far as this, and we'll look at these kinds of things a lot more later on, but basically all that we're looking at here is L is ligand, and here this big blue thing is your P. Now, you can also talk about, any of these other ones being ligands, and we'll talk about more. Lots of proteins bind to lots of things, and so there'll be multiple different ligands. But if you're talking about a dissociation constant, you can be talking about one specific ligand-protein interaction. And so in this case, I would say, let's just count the big blue thing as P and the green thing as L. And I'm avoiding talking about any specific examples here on purpose, because it's supposed to be general. Okay, we talked about delta G's before in terms of that's that energy that you need to have going from one molecule to another molecule. It's a reaction that you have to have taking place. Negative delta G's are spontaneous. Positive delta G's are those which you have to input energy for. One of the really nice things is that there's a very direct correlation between the equilibrium constant and the delta G, that energy that you have to have. And so you can calculate your delta G's from equilibrium constants and the amounts of your products and reactants, or vice versa, you can calculate your equilibrium constant if you know delta G's. So they're completely interchangeable relative to each other. I basically just threw this up here to show that there is this very distinct relationship between the two. I don't expect you to remember these equations. If I have equations, I'll put them on exams for you. So let's now talk about why delta G's and equilibrium constants are all well and good, but basically none of biology would work if that's all we had to work with. What we need are some way of helping out these reactions. And the way that these are helped out is pretty much through the activity of catalysis. Catalysis, again, hopefully from chemistry, all it does is change the amount of energy you need to get from one state to another. And in terms of molecular biology, it's almost always proteins that are doing this. These are the enzymes. All they do is lower this so-called activation energy barrier from one which generally would never happen on a biological time scale to one that can happen with a pretty high degree of frequency. But still, high degree of frequency is relative. The reactions that take place here are still relatively infrequent, but as long as you have enough of these interactions, eventually some of those are actually going to be able to make this transition. You remember that transition, this amount of energy is going to have to now be similar to the average thermal energy which is present inside any organism at any particular given time, and it's just due to the temperature they're actually able to function with. This enzyme catalysis doesn't change the equilibrium, because all the equilibrium is about is where these guys are relative to each other, and it doesn't change the direction of a reaction. And so an enzyme doesn't push a reaction in one direction. What you have to do is have multiple reactions, one after another, in order to get the irreversibility that you need for something like what we're talking about for getting your macromolecules. Once you've started to build your macromolecule, you don't want to be breaking it down again. So you have to have some kind of irreversibility, and the way that that works, this is how it's diagrammed in the textbook, is you have one reaction here which could go either way, but after that reaction has taken place, there's another different reaction that can go in either direction, yet another different reaction that can go in either direction, and then eventually you get down here. This is not going to be able to go back to here. And it's that idea of coupling all of these multiple different, usually enzyme-catalyzed reactions to get you from your starting reactant to final product. How do we do this? Again, mostly talking about, it's all about the water, but we also have to have activated intermediates. Because in general, this molecule is not going to go to that molecule unless we have some way of making a hydrolysis reaction really, really positive, and positive enough, I should say, positive, not, I should say, with a high enough negative delta G in order to get these reactions to take place. So in 95 to 99% of biological reactions, you have some kind of high energy intermediate, usually using one of these carrier molecules. Everybody's favorite is ATP. The ATP, adenosine triphosphate, will add a phosphate to some intermediate, and that intermediate now can undergo condensation and give you these covalent bonds that you need in the first place. You can't go from here to here, even using ATP. You have to go via these high energy intermediates. Now, why do we care about this? And that's because for polymerization, it's exactly the same slide as we had before. You also use these high energy intermediates. That's shown here. Nucleoside monophosphates, which we need to get into our polymers over here. This monophosphate gets turned into a high energy intermediate. A triphosphate actually needs two of these ATP molecules. This now high energy intermediate gets added to this polynucleotide chain, but it turns out that even having a triphosphate here is not enough to get this energetically really unfavorable reaction to take place. You also have to have hydrolysis of these two phosphates in order to get full polymerization that takes place, and also gives you extra irreversibility as far as the process is concerned, because you've hydrolyzed this last bond. So this is what we talked about. Again, all of chemistry in actually more like 40 minutes than an hour. What atoms? All about weak interactions and water, strong interactions, activation energy, and precursor activation. I did want to mention here in particular panels 2.1 to 2.7 in the textbook, which provide also a nice overview in a really condensed fashion of what processes are important in terms of the chemistry. Now I'll talk about proteins. May not get to the end of this, so Eric can talk about them on Friday. He'd be more than happy to talk to you about them. But at least start out with some of the structures in terms of what these proteins look like. Proteins are made of amino acids. These are just 4 of those amino acids up here. They have an N-terminal or an amino terminal end. They have a carboxy terminal end. The acid part here is that they can lose this hydrogen ion and then become stably charged. These are also, you'll notice, I said amino end and a carboxy terminal end. These have molecular biology polarity because they've got one end which is different from the other end. One of the really nice things about these amino acids, of course, is now you've got a carboxylic acid can interact with an amine. This forms a nice peptide bond where you have this C double bond O attaching to N. These peptide bonds then hook up to make a polypeptide. Translation process, again, it's all through these condensation reactions. Once you make a molecule like this, it still has molecular biology polarity. It's got an amino end and a carboxy terminus end. Doesn't matter how long your polypeptide is. It's always going to have an amino terminus and a carboxy terminus. Very often people talk about these as the N terminus and the C terminus because molecular biologists are lazy and they like to abbreviate things. What are those amino acids? They basically come in, I like to think of them as sort of 3 different flavors. It's all about the side chains. All of them have the same carboxylic acid and amino groups, but they all have different what are called side chains that are coming off of this carbon in the middle of your amino acid. There are, again, these multiple different flavors, basically 3 as far as I'm concerned. We've either got these so-called charged amino acids, which we'll talk about in just a second, polar amino acids, and non-polar amino acids. Yeah, question in the back. The actual structures down to the electrons, no. You will not need to know the structures of amino acids. I think they ask you to do that in biochemistry. You also will not be expected to know the genetic code for this class. But what I would expect you to remember is that there are these multiple classes, and the amino acids that we talk about a lot, I would expect you to remember which ones those are. For instance, these guys up here. The negative and positive ones, we'll talk about these in just a second, polar amino acids and the nonpolar amino acids. Nonpolar amino acids basically means they're not going to be interacting with water, so that means they're probably mostly going to be in the inside of your protein. The polar amino acids are going to be interacting with the water, so they're going to be in the outside. And then these really cool and interesting ones that you might want to remember are these which are the charged, and it's really because they're about acidic and basic interactions. So loss of hydrogen, addition of hydrogen in exchange with water. So if you look at start out with the acidic amino acids we have up here at the top, glutamate or aspartate. No, you don't need to know the single letter code, but again, molecular biologists are lazy, so we like to use it. Glutamate and aspartate are those which are generally negatively charged because they're losing their hydrogen atoms. On the other hand, lysine and arginines, Ks and Rs, are basic because they usually are going to have this hydrogen atom which they've picked up from the water. And because of that, these are going to be now stably, positively or negatively charged amino acids that you have in your proteins. So those are going to be interacting either with each other or some other kind of ligand. So let's look at protein structure. There are really 3 or 4 different levels of protein structure: primary, secondary, tertiary, and quaternary. Quaternary is just a bunch of tertiaries being put together. Important here is that the primary protein structure is just that sequence of amino acids from N terminus to C terminus. So as they're hooked up together in the peptide chain. And as we'll see later, you can get this from your nucleic acid sequence because of the genetic code. So primary sequence is really, really easy to get. These strings of amino acids that are attached to each other will then start to arrange themselves in three-dimensional space. And that first arrangement is what's called a secondary structure. These are mostly alpha helices and beta sheets. There are some other ones as well, but those are the major ones. And then these secondary structures assemble into tertiary structures. And it's really that tertiary structure. As I mentioned a little bit last time, whenever I say structure, you should immediately think function, exactly. So it's something that has that structure, and that stable structure, stable tertiary three-dimensional structure, that's what's going to have the function. Many of these proteins interact with other proteins either of exactly the same sequence or of different sequences to give you multiple protein assemblies. People talk about these as quaternary structures. Most of the proteins actually function like this, but we don't know very much about this, so we usually talk about them functioning like this. How do you look at protein structures? Multiple different ways of doing it. This one down here in the lower right-hand side is what's called the van der Waals structure. Remember talking about van der Waals interactions, exactly the right distance, the right place to be in order to be attractive interactions. What people will do here is just basically draw a ball where that just right is. Unfortunately, these kinds of structures down here are kind of really hard to look at and hard to imagine how they're going to be interacting with each other. So most of the time in this class, we're going to be looking at structures like this, which are really cartoons that will have the secondary structures like the alpha helices and the beta sheets. But really what this thing looks like is down here. You can also look at the individual side chains. For instance, here's a nice one sticking out on the side. This looks like a lysine here. Or you can just look at where those alpha carbons are in the peptide backbone. But again, we'll mostly be looking at things like this and occasionally looking at them like this. So what are the secondary structures? Main ones are, again, alpha helices and beta sheets. Both of these were discovered, or at least proposed, by probably Portland's most famous son, Linus Pauling, where he was looking at structures of different proteins, particularly silk and keratin, and came up with this really amazing theory for how these things are going to be fitting together. And basically what he said is that these structures, which you're finding in pretty much all proteins, have to be something that's independent of all of those side chains, has to be about those backbone atoms which are there, and so the interactions between those backbone atoms. And so in the case of the alpha helix, it's that oxygen atom which is part of the backbone together with the hydrogen atom that's hanging off of the nitrogen. These are just hydrogen bonds. They're hydrogen bonds that are forming inside one primary sequence of your amino acids. Don't expect you to remember all these numbers here, but important for this structure is that it's, it's hydrogen bonds that are between the backbones that are holding things together, but more importantly, what does it do to all of those side chains? It sticks them out to the outside, so now they can interact with other side chains or ligands. Yes? Certain amino acids that tend to form helices more than others. Yes, but we're not going to talk about them per se. But yes, they definitely are. And you can make predictions in terms of what's likely to form alpha helices versus beta sheets or not, as the case may be. We didn't talk about proline, but proline's really good at breaking these helices. The other kind of major secondary structure that you find in proteins is a so-called beta sheet or beta strands. And here again, it's just the same thing. It's the hydrogen bonding that happens between backbone atoms, but now instead of the same primary sequence of amino acids interacting with, they're now interacting with different ones on different pieces of your primary structure. So here we have hydrogen bond formation, everything which is in red here, now interacting with your NH, which is on a different strand. This can happen in an anti-parallel sense, where you've got these two associated with each other. An anti-parallel, again, is molecular biology polarity, N-terminus to C-terminus. Or they can be parallel to each other. Makes no difference, and that's what's just shown over here, is you can have either anti-parallel. Here you can have just a short loop of amino acids between your secondary structures, or you can have a longer loop between the two. Now you've got secondary structures. You have to put them together into tertiary structures. That's what's called protein folding. This is all about those weak interactions and the weak forces that are involved in putting things together. You can have your stably charged side chains. Now a negative charge here on one of your acidic side chains, positive charge on a basic side chain. That holds things together. You can have van der Waals attractions here. You can have hydrogen bonds. You can have hydrophobic interactions, all the water molecules on the outside pushing things together. That will now give you your final tertiary structure. This process, going from primary sequence to tertiary structure, is still one of the big unsolved mysteries of molecular biology. We don't know how it works. We would love to, because again, once you got the structure, you've got a much better idea what its function is. But we don't know what those structures are going to be yet. We do, however, know about a few things, and I'll cover those in the last few minutes of lecture here. First one of those is the so-called coiled coil, also known as leucine zipper. Coiled coil is a much better way of looking at that. You remember when I mentioned the alpha helices? They've got their side chains sticking out all around the outside. If those side chains happen to be hydrophobic or nonpolar side chains, where are they going to want to be? Interacting with other hydrophobic nonpolar side chains. So if you've got those, and one of the things that I forgot to mention is that when you go up an alpha helix, it's about 3.5 amino acids per turn. So every 3.5 amino acids, well, you can't do half amino acids. So every 7, which is your integral number, every 7 amino acids, you have a nice hydrophobic residue that's sticking out. It's really nice to be able to stick two helices together. And it turns out this happens very often, particularly in terms of dimerization, but also in terms of some of the long structural proteins, like the keratins, which are what Linus Pauling used in the very first place to discover these structures. Yes?
Every 7 there's what sticking out?
You'll have a hydrophobic amino acid sticking out. Everybody's favorite is leucine, and so very often people will talk about these as the leucine zipper, because each of those are then coming together to interact with each other. Second to last slide, just wanted to talk about domains versus your individual structure. Basically, all that a domain is, and we'll talk a lot more about domains as we move on through the rest of the course. A domain is a separately folded, i.e. separate three-dimensional structure, which is part of a protein. So if it's got a separate structure, it's going to have a separate function. Yes, thank you. So those are going to be fit together. There seem to be only about a couple thousand of these that we've found so far, and we're probably not going to find too many more, except if we look in viruses, which is a whole different story. But this process, again, if we look at one of these, so-called ribbon diagrams over here, the different colors represent different domains, different structures, which are going to give you different functions. And we know that in the human genome and many other genomes, these things get moved around all the time. And so it's a mixing and matching that takes place. What do these structures look like? Basically, alpha helices and beta sheets, or some kind of combination thereof. I'll leave you with this slide basically to give you a bit of an idea of scale. Some proteins are really pretty small. Some are pretty ginormous. And here's a nice double-stranded DNA versus a single-stranded RNA for comparison purposes. That's about the size of a membrane. So have a great weekend, at least as far as I'm concerned, and don't give Eric too much of a hard time on Friday.